那我就给你这个建议，我还是觉得真的挺有帮助的。对，
对，我觉得这个 bottom line 就是美国还是需要最好的 biomedical research，这个是不会变的，是不是？
他们很 confident 的这件
对
事情，因为他们说，其实08年那次 cut 因为很严重嘛，之后很快 NH 的，就那一次是 NH 的 grand double。嗯，对，因为大家 cut 以后总会很快意识到你还是需要做 medical research 的。
对对对。
而且最近这两次的话，好像都是因为川普上台，是吗？就是上一次也是因为他上台，然后马上就要进行 cut，然
对，但是他
后这一次
上去也没有
又是
做成嘛，但这一次当然不太一样了，政治环境不太一样了。对，但是
这这是有帮手的，主要是。
对对对。
就是因为对他们这些很 senior 人来说，他们就是这些事情在我们 career 里面已经发生过很多次了，所以他们你们还很年轻，见得少，大概就是这个意思。但我觉得听完了以后心态很好。嗯，
我不知道你
嗯，
们那边怎么样。我们单位实际上上周开始，他们已经在做，就是如果 cut 到15%的预案了，反正，嗯、他们要按照这个预案来处理，就是未来1、2年我们好像是可以 handle 的。嗯，
那可能每个学校 case 会差别很大。
对，是的，因为我们州政府给了很多的支持，所以呢，他也就是呃尽量的能够，就是说呢，
压力小一点。
能够政府满意比较好一些。
那要不我们那个就准备开始？嗯，我们不
OK，
会拖太多的时间。嗯，好的。
yeah, I can get it started. So, hello everyone. Host. Sorry, that's my dog. Sorry, 1 minute. Okay. Sorry for that interruption. Yeah, today Yan and I will host Dr. Hong Yang's C-Day Happy Hour seminar. So Dr. Yang received his PhD from Beijing Institution of Biotechnology, and then he came to US and then did his postdoc training at University of Utah and then Jackson Lab. So in 2018, Dr. Yang started his lab at University of Indiana and until now. So his research has not been focused on chronic liver disease, and most recent research is on Lincoln RNA biology and then alcoholic liver disease. So since he started his lab, he has been very productive and successful, including many achievements like NI AAA KO1 award and his recent R1 award. And he's very active in the liver disease studies. He's on the editorial board of hepatology, and he was top peer reviewer for both hepatology and hepatology communications in last year. So let's welcome Dr. Yang. Now time is yours.
Okay, thank you. Thank you very much for the great introduction. And I will share my screen first. It said it's not allowing me to share the screen.
I think it should be okay. Can you try share? Yeah, right now the setting is okay.
Uh,
You try one more time?
okay, I probably need to log off and log in
Yes,
again.
I'm
And
logging.
Okay.
that's that's what this said. I needed to make make some changes on the. Sorry about that.
That's okay. Still, yeah, waiting for others join us.
Yeah, we have the same thing since Zihong is not here. If you have any question during the talk, you can type into the chat, and then we will follow those questions after talk finishes.
Good.
Okay, we're good.
Okay, so let me.
Which there. Good.
Good. Okay.
Yeah.
Okay, so thanks everyone for joining this today's seminar, and it's my great a pleasure to introduce our recent work and mainly focus on the long non-coding RNA in the alternative for splicing. And our lab is mainly working on the alcoholic liver disease, and it's also related to the metabolic disease. So it's a great opportunity to present here and I greatly appreciate. And according to the data from United States, there's about 84% of people with aging 18 and older reported that they drinking alcohol at some point in their lifetime. Although the number of all kind of drinking, including the moderate drinking, risky and heavy drinking, it's high in North America, and the population is much higher in Asia. And I think this must contribute a lot from China, especially in male compared to female. So alcohol consumption could be a serious society problem, and also it hurt our health as well. So one of the major problem caused by alcohol drinking is alcoholic liver disease. It ranged from alcoholic steatosis to steatohepatitis, fibrosis, cirrhosis, and eventually the hepatocellular carcinoma. 90% of a heavy drinker will develop steatosis, or we call the fatty liver, which is the earliest stage of alcoholic liver disease. Uh, it has a slightly elevated in the blood level of ALT, AST, and histology of the liver showing numbers of large or small size of lipid droplets in the hepatocyte. It can be reversed by 4 to 6 weeks of abstinence if you are stop drinking. But if you're continue drinking, 20 to 40% of steatosis will develop to the steatohepatitis or fibrosis, which is characterized by the neutrophil infiltration, hepatocyte blooming, necrosis, or cholestatic changes and fibrosis. Eventually, 3 to 10% of cirrhosis will end up with the hepatocellular carcinoma. So after the alcohol's uptake, most of alcohol is absorbed in the stomach or the upper small intestine. After the absorption, alcohol is transported to the liver through the portal vein. And the liver is the major organ which metabolizes more than 90% of ingested alcohol into acetaldehyde through the ADH and CYP2E1 and the catalyze. And acetaldehyde is subsequently converted to acetates by ALDH, which is released from the liver and metabolized by the muscle and the heart. Metabolizing alcohol is will promote NADH accumulation and RS generation, which inhibits mitochondrial fatty acid oxidation and promote fatty acid synthesis, inflammation, and cell apoptosis and necrosis through inhibition of PPR-alpha, AMPK, and active the SRBP pathway. So alcohol consumption is also associated with increase in the intestine permeability and LPS leakage. And this is also leading to the activation of a Kupffer cell in the liver and release the pre-inflammatory cytokines. This cytokine can increase TGF-beta level and active hepatic static cells and leading to increase of fibrosis. So the alcohol also can affecting the adipose tissue and reduce the adiponectin, which reduce the AMPK and contribute to the lipid accumulation in the liver as well. So to study the signature and the mechanism among those different stages of ALD, and my mentor, Dr. Suthaling Ponsko, had collected the whole blood from patients with a heavy drinker and alcohol cirrhosis for years. And those samples were sent for the microarray RNA sequencing and LC-MS for the different RNA profile or link RNA profile and metabolomics analysis for the biomarker screening. And I'm not going to go through all these projects, but I want to introduce specifically for the long non-coding RNA profile. So this is the link RNA profile from the peripheral blood, and one thing interesting is along with the progress of alcoholic liver disease, the signature significantly disrupted the gene number was increased accordingly. So AC1 only has hundreds of DEG, and which means the different expressed link RNAs. And AC3, which is a later stage or severe stage of ALD, has thousands of DEGs. And we detect also confirm those expression change use the real-time PCR and also select several for the ongoing projects. I hope I can introduce those projects later. And H19 also is one of them. It didn't show in here on the list is because we list those link array based on the unique expression in the different stage. So H19 is not uniquely expressed in any one of the disease stage. So it's like a multiple stage will overexpress H19, so it didn't show in here. So as we know, there's only 3% of a human genome could transcript to the protein coding RNA. Large number of transcript is appear to be non-coding RNA. Among those non-coding RNA, there's over 4,000 link RNA were recognized and have the HGNC gene symbol, which is far more than the microarray and other non-coding RNA. So link RNA actually is transcript with longer than 200 nucleotides and not translated into protein. So it don't have the protein coding ability. But its function and very complicated and there's a lot of unknown and didn't being fully studied. So it had been a long history. The non-coding RNA had been considered as the junk RNA. But now it becomes the golden man and clearly left the junkyard behind. And several Nobel Prize had been awarded for discovery related to the siRNA and the microRNA. And guiding RNA is also a short and non-coding RNA in the CRISPR, and it's also very critical and important. And also last year, a few years ago, the RNA vaccine, and that's Nobel Prize is also related to the RNA modification. And however, none of those discoveries, the long non-coding RNA haven't, have any Nobel Prize yet. So, but I hope this field can be studied more, and I think it's also have a very good potential to have some big discovery. And to be noticed, the first long non-coding RNA discovered was linker H19, which is found in the 1989, which is almost 36 years from now. So linker H19 is one of the first discovered linker, as I mentioned, and it is length of 2.3 kilobits. And H19 also is a paternal imprint non-coding RNA, which means the paternal inherent allele is silenced and only maternal inherent allele is active. So H19 is highly expressed in the embryonic stage, but its expression were diminished in the most tissue after birth. But interesting is its expression is upregulated in many diseases, including cancer and multiple liver disease. And we previously report that overexpression of H19 can worsen the hepatic steatosis in high fat and high sugar induced NAFLD. And however, the role of H19 in the pathogenesis of alcoholic associated liver disease is still unknown. And to explore the potential role of H19 in the pathogenesis of ALD, we first detect the expression of a hepatic H19 in patients with alcoholic liver disease and also in the asno-fitted mouse model. And we found a significant increase in the hepatic H19 in patients with alcohol hepatitis and alcoholic cirrhosis, and as well as asno-fed mouse liver. And the bottom panel is the RNA in situ staining for the H19. It's called RNA scope. And the green staining indicates H19 is positively expressed mainly in the hepatocyte. And we also have the co-stain of H19 with HN4 alpha and indicated they can overlap with HN4 alpha positive hepatocytes. So to test the H19's role in the ALD, we used both gain and the loss of function approach. And in the gain of function approach, we used the adenoma-associated virus subtype 8 and with the TBG promoter to specifically overexpress H19 in the mouse liver. And then we feed those mice with alcohol plus 1 binge, and which is famous as AAA or gall binge model. And we feed in the control diet for 5 days and then follow the 10-day either control diet or SNO diets for 10 more days. And at last day before, an hour before the harvest, we binge the alcohol and to boost the liver damage. So we observe the overexpressed H19 can worsen the hepatic steatosis in the asno-fed mice, as indicated by the HE staining. And liver injury will also increase as indicated by the plasma AST level. So this data suggests that H19 can promote alcohol-induced hepatic steatosis and liver injury. So as the next step, we use the loss of function approach. And since H19 is a paternal imprint genes, in which only the maternal inherent allele is active, so we generate H19 maternal specific knockout mice to knock down H19. So these mice were subjected to the ASNO plus 1 binge model as we described before. And the hepatic deletion of H19 in the H19 maternal specific knockout mice were confirmed by the real-time PCR. So in contrary to gain of function approach, and the loss of H19 can protect against alcohol-induced hepatic steatosis. And L-red O staining also used to stain the lipid accumulation in the liver. So we have summarized in a review paper and for the possible mechanism of how H19 can be functional. And the first H19 is the host gene for microRNA 675. So H19 expression will co-express the microRNA 675 and it's leading to regulation its target gene and protein levels. And the second is H19 also can function as the sponge for other microRNA, like LED7, microRNA 106, or 29A, and others to block those microRNA's functions and to inhibit their target. And H19 also can interact with the proteins, depend on the function of the protein, and it can be functionally regulate chromatin and methylation status, and like through interact with the G9A or regulate the RNA stability, looks like through the interaction with the RNA binding protein. So to study the mechanism, we first perform the RNA pull down, use the built-in labeled sense or antisense H19 RNA. A unique band in the sense length and were cut and sent it for the MS. And a total of 48 protein were discovered, and a total 6 of them were selected as shown in blue for the validation. But only the PDBP were showing the positive binding. So PDBP is RNA binding protein that belongs to the subfamily of ubiquitous expressed heterogeneous nuclear ribonuclear protein. So it's also called HNRNPs, which can recognize and interact with the enhancer or silencer to act primarily as a repressor for the precursor mRNA alternative splicing. So alternative splicing is a process of selecting the different combination of splicing sites within a pre-mRNA to produce a variable spliced mRNA, which can improve the gene coding efficiency and contribute to the protein diversity. So since the alternative splicing factor changed in the alcohol cirrhosis, next we ask whether this alternative splicing event can be changed as the consequence. So from the RNA sequencing analysis, we found an increase in overall alternative splicing event in patients with alcoholic cirrhosis, especially in the category of alternative exon and exon skipping. Since H19 can bind to PDBP and PDBP is the alternative splicing factor, we then ask whether H19 also can regulate alternative splicing. So interesting is we found the H19 overexpression also can increase alternative splicing overall events in the alcohol-fed mouse liver, and especially in the same category of alternative exon and exon skipping. So there's another interesting thing actually when we are performing those experiments, and we did another project which is analyze the RNA sequence from a peripheral blood RNA sample for the regular mRNA profile from the human healthy controlled patient with alcohol cirrhosis. We then observed that the number of different expressed mRNA and it was also like increased alongside with the disease severity. So similar like the linker profile. And when we do the pathway analysis, indicated those differentially expressed gene in the AC2 and AC3, which is a later stage of alcohol cirrhosis. And the pathway were enriched in the alternative splicing pathway. And when we do the heat map, we found there's one of the most significant changed gene is PDBP1. And the RAVER1 actually is another interacted protein with the PDBP1. And also there's another the alternative splicing factors, including SF3B2, also were showing the same pattern as the PDBP1. So to identify the alternative splicing target of H19 and the PDBP axis, we first examined because we want to know the target needed to be functional related to the function we observed in H19 overexpressed mice. So we first examined the metabolic data and found a significant reduction in the hepatic methionine level in the mice overexpressed H19. And methionine metabolism is a completed pathway involving several key enzymes in the methionine synthesis. So we examined the genes in the methionine metabolism pathway and found that the expression of BHMT was markedly reduced in both ethanol-fed H19 overexpression mice and in the AML12 cells, which is the mouse hepatocyte cell line, and uh, and also in the mouse livers. And and as shown in the pathway, the reduction of BHMT also can lead to the increase of homocysteine and a decrease the methionine and SAMIs. Interesting, the induction of SAMIs and methionine had been shown to be associated with alcohol-induced liver injury. So we next the question is asking whether BHMT undergoing the alternative splicing. So interesting is a previous study showing a splicing variant of BHMT leading to complete loss of its function in the hepatocellular carcinoma and HepG2 cell line. So use the same primer, we found the same BHMT splicing variants in the livers of a patient with alcoholic hepatitis. And in the mouse, BHMT has two splicing variants, BHMT201 and BHMT202. And from the RNA sequencing analysis, we found the BHMT201, which has the protein coding ability, was reduced, while BHMT202, which has no coding ability, was increased. So our data suggests that H19 inhibit the BHMT protein through switch from the protein coding isoform BHMT201 to non-protein coding isoform BHMT202. So from the consequence of this reduction of BHMT protein level, and the methionine level was also reduced in H19 overexpression mouse liver. And the increase of H19 and this methionine level was also increased in the H19 maternal knockout mouse liver. So to investigate whether H19 regulated BHMT splicing through PDBP or whether PDBP1 could lead into BHMT alternative splicing, we generated a stable knockout down PDBP cell line and found the knockdown PDBP could promote BHMT form the BHMT202, which is retained intron and lack of protein coding ability. And a stable overexpression of H19 in this cell line also leading to the same consequence. So furthermore, we use the PDBP antibody to perform an RNA immunoprecipitation and found the PDBP could positively bind to BHMT mRNA, and this binding were inhibited by the alcohol feeding, and H19 overexpression could further inhibit its binding. So the last question we ask is whether restoration of BHMT could rescue H19-induced hepatic steatosis. And previous work indicated that knockout BHMT could promote alcohol-induced steatosis. And for the gain of function approach, we overexpress BHMT used AV8 with TBG promoter and found that restoration of BHMT can rescue H19-induced steatosis in alcohol-fed mouse model. So in the summary, an alcohol consumption increased the H19 expression, and H19 could interact with PDBP and dysregulates alternative splicing events, in particularly the BHMT protein isoform were inhibited, and leading to reproduction of methionine and SAMIs, which contributed to the development of alcoholic-associated liver disease. So there's also multiple paper had been focused on the H19's role in the alcohol in the multiple liver disease. And like I introduced, the H19 were first actually get to our uh, attention is because when we do the SHIP knockouts and we found the SHIP can interact with BCL2, and then through SHIP regulate the H19. And then we found H19 interact with PDBP and then regulate SREBP1 cleavage and downstream fatty acid synthesis targeted genes. And Dr. Huiping Zhou, her group is also did a lot of study on the H19's role in the cholangiocyte and found it can regulate the bile acid pathway and also can interact with another RNA binding protein called HUR. And after the interaction, it can stabilize the H19 and then package it to the exome to release the and release the to outside of the cell and active the macrophage activation and hepatic static cell activation. And there's also another paper used the BDL model and found the H19 also can regulate the ZEB1 regulate APOCAM expression and regulate the cholangiocyte proliferation. And when we do the RNA scope, and we found H19 also expressed in both hepatocyte and other cell types, and also very close to the cholangiocyte. And we are assuming they may be also expressed in the progenitor cell and evolve in the liver cell damage repairing and or the progenitor cell proliferation. So those could be our next step. And the second thing is we also generate the PDBP flux mice and also the hepatocyte specific knockout PDBP and subject to the alcohol model. And we hope we can present those part of the data in the future. And I have second part we just recently finished. It's related to another gene called FOF2, and it was just released probably yesterday in the online in the hepatology communication. So this project is from the GWAS analysis, and there's a genetic predisposition of alcohol-associated liver disease, and it called the Genome ALC project. It recruited over 1,000 patients with alcohol-associated cirrhosis from 6 countries, along with the 600 heavy drinkers without known liver disease, and to identify the common genetic variants underlying the risk for the alcohol-associated cirrhosis. GWAS reviewed the significant associated SNP on the chromosome 5, and with the most significant SNP is located in the intron of FOF2 genes. So FOF2, also known as UBXD8, is a ubiquitin ligand adapter protein that plays a crucial role in the degradation of misfolded protein in ER and lipid droplets, and primarily through intact with the P97 and also called the velocin contained protein and VCP. And recently, research also showing that the FOF2 can interact with iron SIG1 and regulates the cleavage of SREBP1, excuse me, which is a key factor in the controlling fatty acid synthesis. So to investigate the FOF2 role in the alcohol-associated liver disease, we first examined its expression using a well-established chronic alcohol-fed plus 1 binge model, which we just present in the H19 project. And both real-time PCR and Western blots analyze confirmed that alcohol feeding can increase the FOF2 mRNA and the protein level. And when we analyzed the isolated primary hepatocyte and the non-parenchymal cells, we found that increase in the FOF2 is primarily in the hepatocyte, but not in the parenchymal cells. And this observation is also consistent with the data from a single-cell RNA sequencing in the human liver atlas, which is also indicated that the FOF2 is mainly expressed in the hepatocyte. So, furthermore, to confirm the induction of a hepatic FOF2 in the disease condition, especially the alcoholic associated liver disease, and we analyzed Dr. Bingal's recently published database in GCI. And we add a more healthy control by including the CD45 negative cells as well, and replot the figures. And then we found the FOF2 was increased in the both alcohol cirrhosis and alcoholic hepatitis samples, and mainly in the hepatocyte population. So to detect whether knocking down FOF2 could prevent alcohol-induced liver injury, we selected the most effective the shRNA from the three candidates and clone it into the AAV vector and package it into the AAV subtype 8, like we did for the H19, and to specific the targeting the liver to knock down FOF2. So 7-week-old wild-type mice were injected with the virus through the tail vein, and 2 weeks later, they were subjected to the alcohol feeding model, and both MR and the protein level of FOF2 were successfully reduced in both control and ethanol-fed group. And this is confirming the knockdown efficiency in the liver. And additionally, we observed the reduction of liver to body weight ratio in the ethanol-fed group. And FOF2 knocking down in the ethanol-fed mice significantly reduced the hepatic steatosis, and as the evidence by decreased lipid droplets accumulation in both HE and red O staining. So to further investigate the mechanism underlying the protection role of FOF2, we conduct RNA sequencing analysis on the ethanol-fed mice livers. And in the FOF2 knockdown group, we identified the 538 upregulated and 198 downregulated genes compared to the control. And gene set enrichment analysis revealed that the top enriched pathway were related to the fatty acid metabolism, including the linoleic acid and retinol metabolism. And notably, the heat map confirmed the FOF2 as one of the most downregulated genes, and further confirmed the knockdown efficiency. And additionally, gene involving alcohol and fatty acid metabolism were also significantly affected and highlighted by these orange arrows. So we then utilized there's a database called the TRUST database to identify the targeted gene which is being regulated by specific transcriptional factors. And we identify SREBF1 is one of the key regulator for the fatty acid metabolism and emerged as one of the top transcription factors. And the heat map showing that the genes targeted by SREBP1 were reduced in FOF2 knockdown group. A recent study had indicated knocking down FOF2 could significantly reduce the cleavage of SREBP1 and its transcriptional activity. So additionally, PCX9 is another key gene in the lipid metabolism, also significantly reduced. And previous studies suggest that the PCX9 deficiency can reduce hepatic steatosis by regulating the turnover of low-density lipoprotein receptor, and thereby affecting the LDL cholesterol balance. So we confirmed that a reduction of PCX9 in both MR and protein level And as expected, the plasma PCX9 and LDL level will also decrease. And lipid droplets accumulation is a delicate balance between fatty acid uptake and synthesis, and also the degradation, which including the beta oxidation and the lipolysis, and regulated by the ATGL, CGI58, and the EMLD2. And while the mRNA and the protein level of ATGL itself didn't change, and its activator CGI58 was upregulated in the FOXP2 knockdown livers. Additionally, lipids activity reflecting the ATGL enzyme activity was also increased in both the whole liver and isolated lipid droplets. And notably, ELMOD2, which inhibited the transport of ATGL from ER to the lipid droplets, and was reduced in the FOXP2 knocking down livers, further enhancing the ATGL activity and promoting the lipid breakdown. So, in a quick summary, and FOXP2 knockdown in the liver increased the CCI58 and reduced the ELMOD2, which promote ATGL activity and lipolysis, and thereby decrease lipid droplets accumulation and hepatic steatosis. Additionally, in the fatty acid synthesis pathway and FOXP2 breakdown, knockdown leading to the reduction of SRABP1 and its target gene, further mitigating steatosis. So consequently, plasma PCSK9 and LDL levels were also lower. And our findings suggest that targeting FOXP2 holding permits for developing novel treatments aimed at reducing hepatic steatosis, particularly in the context of alcoholic liver disease. So I would like to thank my mentor, Dr. Stalin Pascu, and he's my mentor for my K award. And after I have the R1, but I still in the Indiana and collaborating with Dr. Suta. And also thanks to all my lab members and all the fundings from IU and also from AAA. And thanks again. And so this picture is showing the Indianapolis. I don't know how many people had been here before, or no one had been here. Before I moved to here, I thought it's just like a Central of America, not nothing surprise. But after I came here and it's had been already 7 years, it's time is just flying, and this place is also very beautiful. And I hope if there's any chance and you guys can come and visit us, and I can show you around. And Indianapolis is also a very beautiful place and good for living. Okay, thank you very much, and I will take your questions, and if there's any, thank you again.
All right. Thanks, Joe, for your exciting talk and your data. All right. Let's move to the Q&A session. Now, we would like to ask anyone, if you want to ask a question, please unmute yourself and turn on your mic, and then director ask questions. Hey, Doyin.
Hey, man. Hey, John.
Hi,
Long
Josie.
time no see. Let's see a
Hi.
few months ago. Not long. Hey, I always interested in your alcoholic liver study. And I just have very, because I'm not working on, alcoholic liver. I just ask a very naive questions. For the alcoholic caused steatosis, so because your data also showed downregulator SRBP. So if we knock out SRBP or just scab, so what's the phenotype? Is risk everything, or because in a fatty liver, like half diet, you basically you can do that. How about in an alcohol setting?
So we didn't do that yet, and we but this just the phenotype we observed.
Mm-mm.
Like SRBP will goes down and its target genes also goes down. But I think alcoholic liver disease a little bit different with the NAFLD.
Yeah,
So NAFLD normally the SRBP will goes up if you block it and it's a beneficial
yeah,
effect. But
yeah.
in alcohol it's a balance. It's not only the fatty acid synthesis but also the fatty acid breakdown. So the in my opinion or in my experience, it's more like the fatty acid breakdown will play a major role, or it was disrupted by the alcohol mostly. And the synthesis will be inhibited actually by the alcohol feeding,
Oh.
or unless there's other factor or long term without a binge. So after the binge, what we observe the actually the fatty acid synthesis is more like already being inhibited.
Mm.
So if
So
at that time when you inhibit SRBP, I'm not very sure, and I think Dr. Mengwei Zhang will be
yeah,
a better person to answer this question, and she is very expertise on this field and also in the SRBP one, and
I know, I
she.
know. We will send she not he. We will send her the scalp mask. Yeah, but I'm not sure. Not for alcoholic project for others.
Yeah, but one thing I can tell is the SREBP
Mm.
will be a little bit different in the alcoholic liver disease and NAFLD or all these fatty acid synthesis. And we are still trying to understand more. And but we can share any updates if I found any.
Then follow you just this question. You just mentioned the fatty acid breakdown seems more important. So even for that like PPAR alpha agonist, although in the human trials it still take time to go, is not great. But in mice
Mm,
still very helpful in the alcoholic in the NAFD. So how
mm.
about PPAR alpha fibro drug in the alcoholic disease?
Well, I'm not sure.
Because
And
it's break down the lipids, right?
yeah, yeah, it should be active for the PPR alpha and then should have the beneficial effect. But
Yeah.
there's one thing very interesting is, we have another project is CPG. It's a CYP4A and 4A10, 4A14, and all these CYP4A genes. And those CYP4A genes will be regulated directly by the PPR. And it will induce during the fasting stage, and PPR alpha will active those CYP4A significantly. But one thing is interesting is when you break or inhibit these CYP4A genes, it's protecting alcoholic liver disease. So it's
Interesting.
very
Yeah.
confusing. It should active, right? The PPR alpha is beneficial. You active CYP3 and help with the omega oxidation. And those oxidation will break it down the fatty acid. It should be beneficial. But actually, in our data, we saw it's if you are inhibit CYP3, it's beneficial. If you active them, it's like a contributing to the fatty acid accumulation, like alcoholic liver disease. So, in the alcoholic liver disease, there's a complicated pathway regulating. For my understanding, is those CYP3 is not only it is regulated by the PPR alpha, but in addition of the fatty acid oxidation, and they have other roles in like the ER stress or something.
Mm.
And
Mm.
so during the ER stress, and this CYP3 also goes up, and it's also contributed to the ER stress. So if you block that, you block ER stress, and you block the stress, and you block the alcohol induced the liver injury. So that's why we see that phenotype. So it sometimes it's not a pure fatty acid metabolism. It's a combination of multiple pathway. So that's
Interesting.
yeah.
Yeah. One more thing, maybe not a question. Also consider the lipid toxicity because significant and break it down. And also that's also connected to your ear stress. Yeah.
Yeah.
Thank you.
Yeah. Thank you for the questions.
All right, Dongyin, thanks for your question. Um, anyone has questions?
Yeah, let me
Yeah,
ask a question. So,
yeah,
Zihong, we talked about your project earlier,
Yeah, yeah,
and
yeah.
uh, looks like H19 is kind of upregulated in many chronic
Yes.
liver disease. So,
Yes.
do you have a sense in general what's the main mechanism controlling its upregulation? Is there, I think you proposed like transcriptional mechanisms, but
Yeah.
is that like stability or other kind of regulations?
Yeah. There's two parts. I don't know which one you mean. It's how H19 were upregulated in the disease stage, or how is H19's function during this stage?
Yeah, if you can comment on both sides, it would be
Okay.
great because
Yeah.
like you follow up
For
like several studies, all right? So like they have
yeah.
different roles in different disease settings.
Yeah. Yeah, for the second part, for the role of H19, actually, I just showing one of the summarize and in the different cell type, including the cholangiocyte, hepatocyte, even progenitor cell, and also in the macrophage and stellate cell. And for the upper stream, like how this H19 were increased in the disease stage, I didn't mention too much. So I will just focus on that part. And there's a several way to upregulate H19. First is the cell type specific. Like H19, normally there's no expression. So during the stage, during the disease stage, and this H19 significantly increased, especially when there's a cirrhosis stage and also later stage of the liver disease. And the one of the reasons is because those high proliferate the cell or accumulates, and those cell express the highest H19. And so that's why you see H19 work goes up dramatically. And this is overall for the cell type difference. The second is in the cell itself, in the inside of the cell. And it can be regulated by the multiple mechanism, including the methylation level, because H19 is a imprint gene. Normally, the promoter will be methylated, so it's only expressed in the maternal allele, and it's only expressed in the methylation status where reduced, so this gene will be goes up, and then the transcription factor will regulate H19, but it is still mainly regulating by the methylation.
Okay.
And yeah, yeah. So we will work on that part as well. And yeah, but I don't show any data yet. Yeah,
Oh, great. Yeah.
yeah. Thank you.
thank you, Xiangbo. Hey Joe, I have
Hi.
two simple questions. One is related to your model. So
Yeah,
my feeling is like in human, maybe you need like years after probably
yeah,
have a drink, but your animal model looks like it's pretty short. So is it like a general people use this mouse model to induce
yeah.
the liver, like alcoholic liver, right?
Yeah, that's a very good questions, and I just saw from WeChat. I think
Mm.
recently Dr. Wen Xingding
Yeah,
and his
the
group
liver guy.
published a review paper and
Uh,
talking about all the alcohol mouse models,
yeah.
and it summarized very intensively. It's a very good one. I can find it out and forward to you as well.
Yes, please.
And why this model has more dramatic effect compare, normally like before Dr. Bingal's model, we call it Dr. Bingal's model. And before that, we need to feed in the mice with liquid diets for at least 2 months.
Yes.
And you still have a very mild ALT, AST, and also steatosis. But
Mhm.
Dr. Bingal's model, there's one change, is very dramatic, is the binge. So you
Yes.
have to binge drink in the mice, and you can just feed like a 7 or 10 day, and as short as a 10 day, and then you can just oral grudge with the 35% of alcohol, And then after 9 hours, the ALT/AST can reach to more than 100. And also by that time, the steatosis can be significantly observed.
Yeah.
And second, because we did this model actually many times, and that's just a personal experience. And when we collect the tissues and we collect different loop,
Yeah.
so there's a different parts of the liver, right? So normally people just collect the biggest one. The biggest one actually is the male's phenotype. So
Yeah.
if you collect, there's two small, upper small parts, and that one you can see easily has more inflammation and also more steatosis.
Hmm.
So the different loop of liver also showing different stage or damage from the alcohol drinking. And the second is you know the model I also considering is alcohol model cannot be pure alcohol, but I
Yeah.
think it's still immature theory, so I'm not talking too much about this. But this binge drinking will significantly help with the observation of a better phenotype. Yeah.
Okay. Yeah, great. I think I already learned something. Yeah. So the second question is follow up. Say, well, if it is possible that you can have some human sample to test the model, because I believe if in a mice, I think it's pretty fast. So you see these gene changes, but in human, maybe it's better if you can replicate the result by using
Yeah,
human.
that's a very good question. And when we collect those samples, and I had been collaborating with my mentor, and he has collected a lot of human samples,
Okay.
most of the blood samples. But
Yes.
liver sample is more difficult because if a healthy person, they just drinking alcohol, they won't allow you to do the biopsy or,
Yes.
whenever you do the biopsy or you have the sample is most of the later stage.
Hmm.
So alcohol, if you want to induce to the later stage, especially if you want to induce the fibrosis, it's very difficult.
Yeah.
Even the binge model, you won't have any fibrosis. So that's why I said it's immature thought. Is I like the idea of alcohol is a second heat.
Hmm.
Like you need a second heat to boost that damage. And if you are combining with the alcohol with other models, for example, the CCL4, and regularly if you are inject the CCL4, you need 2 months to induce a significant fibrosis, right?
Hmm.
But if you are combining the alcohol with the CCL4 only can use a very low dose, like only 10% of the regular CCL4 dose.
Yeah.
And also, even the low dose, you need to be very careful for during the feeding.
Mm.
And normally, it won't last that long. We just feeding the mice for 1 month, and I injected the CCL4 only like 4 times.
Yeah.
And the male mice, even after just one time, the male mice can die and has a significant damage on the liver.